to bring together the 1,239th meeting of the Entomological Society of Washington. Welcome, everyone. Mr. Secretary, could you please read the minutes from the last regular meeting?
God.
Got it. The 1238th regular meeting of the Entomological Society of Washington was called to order on the 5th of October by President Matt Buffington at 7 WG 33 of the National Museum of Natural History. 21 members and 5 guests were present. 24 virtual visitors also joined the meeting. Recording Secretary Gary Hevel read the minutes of the annual banquet and the May meeting. There were 2 suggestions to better reflect the constituency of the family bird community. The minutes were approved pending this particular change. President-elect Don Weber noted that the committee for officers for next year is hopeful to find candidates for the positions of editor, curator, and president-elect. Members should contact him with suggestions of people for these positions. John also noted that the national meeting of the Entomological Society of America will be held in Oxon Hill, Maryland in early November, and that he and Matt Buffington will be leading interested ESA members on a tour of the site of the CV Raleigh House on Thursday, the 9th of November, after the ESA meeting. The cost of the tour will be $75, last approximately 4 hours, and will include a free copy of Don's definitive book on the life of Raleigh. Floyd Shockley also has a number of tours scheduled during the ESA meeting. Don Davis noted a new book edited by David Grimaldi titled The Complete Insects, which includes a chapter of Don's son Stephen and colleagues. President Buffington announced that Treasurer Abby Kula has received a long-awaited response from a lawyer, and the society should soon be able to engage in investment of funds. Program Chair Alan Norbaum noted that Quentin Wheeler has a podcast called Species, in caps, that recently featured news that Bob Copeland in Kenya has named 41 new insect species. ESW member Brendan Boudinot noted that a new fire ant in Sicily has been described with 19 colonies discovered. Don Weber noted that he has been in Portugal recently, attending a biocontrol conference. Research reports that will appear in ESA concern controls of noxious plants by insects. Manon Davis passed around the latest ESW issue for proceedings of October 2022. She noted that several well-known authors are listed on the cover, including John Burns with 2 articles. Manon points out that John Burns is the most senior of scientists at the Department of Entomology at the National Museum of Natural History. Program Chair Alan Norbaum introduced the speaker of the evening, Alan Cabrero, an NSF-sponsored postdoc working with Torsten Defoe. The title of his presentation was Microbiomes, Phylogenetics, and Heat Shock Proteins of Leaf Life. His background studies were at Berkeley. Bombellidae are a diverse family with some 5,000 known species. Many dwell in deserts. They are nectar and pollen feeders and are best collected during summer, during days with sunshine. Alan traveled with Torsten Defoe to South Africa for a collecting trip and found that Torsten had great skills when climbing sand dunes. His research involves analysis of proteins combined with standard phylogenetic approaches. In the past, leafly workers have experienced frustrations with the taxonomy of Lorditus, a genus known for a long pile on the abdomen. Attention to the antennal segments by earlier taxonomists was inconclusive. Alan mentioned that specimens of this genus are collected individually by netting and that there is no known taxonomic value in the pile. Bee fly larvae are protected by microbes, and this aspect can be used as a tool in comparative analysis. The discussion period after presentation brought forth many questions before adjournment at 8:18 p.m.
Are there any additions or corrections to the minutes having been read?
Yeah.
You mentioned something about Bob Copeland.
Yes.
So he has, he sends out emails. 1 a week on a different species.
Uh-huh.
Yeah, it's true. The notes said he's described 40 species, but which he probably has, but this is a little bit different.
Okay, I'll check that.
I can chat with you.
Okay, thank you.
Either additions or corrections.
There's a Ray Gunn who has his hand up in the chat.
You,
Ray? I think he's unmuted now.
Hello,
yeah, we can hear you.
this is Ray. The oldest member of the entomological, of the Smithsonian Department of Entomology, I believe, is Don Davis. Did you say?
In fact, I think she was bragging. I don't know. I'll check it out.
Yeah.
Thank you, Dr. Gagne. Any other additions or corrections? If not, I have a motion to have the minutes accepted as read.
So moved.
Second. Thank you, Recording Secretary Hevel. President-elect Don Weber, would you like to share your thoughts?
I've done that too much, as according to the last minute. But I do have one announcement. There is to call your attention to the 2023 AAAS Charles Valentine Riley Memorial Lecture. These are usually big picture things, and they usually are not entomologists. And that's true this year. The title is Reimagining the Arc to Impact by Dr. Joe Cornelius, CEO at the Bill and Melinda Gates Agricultural Innovations, and that is called Gates Ag One. And the only thing I know about them is their agriculture, and they probably have a lot of money. The annual AAAS Charles Valentine Riley Memorial Lecture was established to honor Charles Valentine's Riley legacy, blah, blah, blah. You can register for free, and I believe Alan has already sent out the link. It is 2 weeks from yesterday at the AAAS headquarters, which is worth the trip. You know, not even to mention the good appetizers you're going to get at the reception afterwards. The registration or the doors open at 4:30. It's at 5 o'clock, November 15th. And if for some reason you did not receive the email, I have one extra copy. Any questions about that? He will talk. There'll be a panel that has a Q&A session with Sudip Parikh, who's the CEO of AAAS, and it'll be a fun time. Probably even be some good appetizers, and it's free.
Is it in DC?
It's in DC at the AAAS headquarters, which is on New York and 1200 New York Avenue. Yeah.
What's the closest metro stop?
Yeah, there is a close metro stop. I think Chinatown, maybe. Yeah.
That's Metro Center.
Yeah. Well, whatever. It's close. 1200 New York. It's not far away from the metro.
Okay, before we move on with any more officer reports, it's my duty to inform you. Brendan Boudinot will not be here tonight. I apologize. He's horrendously sick. And a sleight of hands that happened this morning. His incredible wife, Jill Oberski, has offered to give the talk tonight. Thank you very much, Jill. Brendan will join us in December because Luke Kreslin has agreed to bump his talk to March. So that's the new order of events. So surprise! Everyone here probably recognized Brendan's not here, or at least you hear that he was not here. Everyone in TV land, sorry, but join us again in December. Jill's going to give an incredible talk, and I'm super excited about it because she's actually focusing on something I'm actually interested in. So I'm very excited. Okay, now back to the regularly scheduled program. Elizabeth Young is not able to join us, but she emailed me that we have 2 new members. Jeffrey Sosa Calvo is one of them. Thanks. It's been a while, yeah. And Davide Del Pol. I just know him by Davide. So Davide, if you're out there, welcome to the society. I'm glad you're with us. Those are the only 2 members as of right now. There's no treasurer's report because Abby Kula's not here.
Wait, no, but they also have to ask about the guests.
Yeah, I gotta finish the officer reports first. Thank you, past president tomorrow. Would you like to present something? Okay, so who else? We've got custodian, Talita. We've decided that we've assigned Talita to be the current custodian, and she'll be voted in hopefully next meeting. And that brings me to the next major point. Normally, as of right now, we have a slate of candidates for all the open positions. Because of the way the universe is operated, we do not have a slate at the moment, but we will have a slate that people can vote on at the next meeting. So stay tuned, and to follow in Secretary Hevel's minutes, please let us know if you're interested in being an editor of the prestigious Proceedings of the Entomological Society in continuous publication since 1884, or if you would like to be considered for president-elect of the society. Those are the 2 positions that we have open. Toledo's pretty keen on custodian, so if you want to run for custodian, you're going to have some stiff competition. Okay, I think that's it for those announcements. Okay, visitors. We have a lot of visitors tonight because of the Insect of America meeting, which is like a sister society to the Insect. I would love to hear everyone here that's visiting to please raise your hand. Don't be shy. We all know who you are, and say who you are and what you work on while you're here. Beulah.
Hello, everybody. Thank you so much for allowing me to attend this evening. I'm very excited to be back in DC. I should just swivel, right? Swivel.
Or just keep turning.
I'm a senior curator at the Natural History Museum. I work on carabid beetles, but I'm also curator of orthopteroids, which I know nothing about. So if there's anyone here that does know about orthopteroids, please speak to me. And I'm also chair of the Coleoptera Society of Great Britain and Ireland, which is a relatively new society that was a creative and desperate endeavor that came out of the COVID pandemic. So that's me. Thank you.
And for those who don't know, the Natural History Museum is the natural history.
Smithsonian is the Smithsonian.
It's the side. So that's the British Museum of Natural History, as we grew up learning. It's now the Natural History Museum. So Bob Anderson.
I'm Bob Anderson from the Canadian which you have heard of, but it's the National Museum of Canada in Ottawa, and I'm interested in Curculeonid weevil systematics, work with Lourdes on a variety of projects, and I'm here visiting the collection and then heading over to the Entomological Society of America meetings coming up. I also will put in a word for Quentin Wheeler's blog, or not blog, but blog and podcast. It touches on a lot of topics that are of current relevance and interest to insect taxonomists throughout the world.
Last
And he's
episode.
a wonderful, yeah, it's great.
Yeah.
He's a wonderful, I think, spokesman for entomology and for collections and for taxonomic biodiversity research. It's really worth listening to.
Andrew.
Thanks, Matt. My name's Andrew Smith. I'm also from the Canadian Museum of Nature. I'm an expert in scarab beetles, so I'm having fun for a few days before the ESA meetings, going through the scarab collection and the type collections. So I'm enjoying being here. Thanks.
Hi, good evening. My name is Pat Bouchard. I'm at the Canadian National Collection of Insects, which is not the same as the Canadian Museum of Nature. I think we have the better. But I work on weevils and tenebriones. I'm a taxonomist. I'm here looking at the collection and also attending the ESA meeting coming up next week. Yeah, no more in a while, so chat about tenebriones.
I'm just Chris Canizaro. I'm just a guest of one of your newer members, David Hewitt.
Excellent.
Yes, I'm David Hewitt. I work in housing and public health. I used to work at HUD. I worked at FDA now. And I was gonna say I recently moved to DC. I moved here 4 years ago. COVID feels like just last year. I worked at the Academy of Natural Science. That's my museum.
The Academy.
Anyone else? Sorry. Yes.
Second meeting. My name is Jeff. Thanks for having me again. Local artist from the George Washington Master's Program. And yeah, here as a sponge soaking up information into illustrating all sorts of bugs. So I appreciate the opportunity to listen and a lot of your knowledge. So awesome.
Am I a guest? Technically, I'm not a member.
Have you been to a meeting before?
Not a
Been
member.
I've been to 2 banquets like a while.
Just
Okay.
introduce yourself.
Oh, sure.
So you should introduce yourself.
Well, and then join.
Yeah,
And then join.
yeah. I only have Romanian money in my wallet. I haven't transitioned back. My name's Luke Crestline. I am a new postdoc in Hymenoptera at the Smithsonian, and I work on little tiny whitefly and scale parasitoids, things that I think are very cute and everyone else thinks are less, but they're useful. They're very useful.
Yeah.
Luke is an academic cousin of mine. He actually started here as a intern and then went to Riverside, get his PhD with my former advisor, John Parody, who will be here tomorrow. We have to clean up.
I know.
I know. So I have a soft spot for Luke. Great to have him back.
I owe a lot to Matt.
No, you don't. You owe yourself.
Maybe maybe.
Are there people online that want to say hello? Do anyone want to introduce themselves? Does anyone want to introduce yourself out there in TV land? Okay. Excellent. Okay, we can move on to the presentation of exhibits, which is kind of show and tell. Does anyone have any exhibits they would like to share with the group?
Oh, I could ask you 7 things you don't know about C.B. Riley. I already exhibited it. This is the book. There might be 1 or 2 spots still left in the ESA tours, which is on Thursday. You would not sacrifice 1 day of the meeting if you came on Thursday because it's afterwards. It starts at 9 o'clock in so-called National Harbor. And 7 things you don't know about C.B. Riley. And I'm gonna ask, he was a promoter of Rocky Mountain locust as blank. Come on. Food, thank you. Yeah, he had the St. Louis chefs cook it up. I mean, this is entomophagy before
it was cool and hip.
Well, I guess he was trying to make it cool and hip. And here's the last question. Was considering, seriously, taking a position as head entomologist of what country when he died at the age of 52?
I'm going with France.
Kiwi, baby. Yeah.
Was it a country still?
Yeah.
Wasn't it?
I think it was actually still. No, I think it was independent. Anyway, he was.
Just
But he didn't get an opportunity because he crashed his
Empire
bike.
didn't have.
Where we're gonna go see in Calorama
Yep.
next Thursday.
Okay, I did fail to bring 2 exhibits, so we'll have to do that next time.
Yeah, you will be able to show.
Show this kind of things. Okay, I think with that, we're ready to move on to the presenting of fact figures. And I'll hand that over to Program Chair Alan Orbaum. There's gonna be some figures.
Always.
Yeah.
Well, okay, I have the pleasure to introduce our speaker tonight, Dr. Jill Oberski, who's kindly standing in for Brendan. Jill is a postdoctoral visiting scientist at the National Museum of Natural History. She completed her undergraduate education at Malachester College in St. Paul, Minnesota, where she started her research career on the systematics of Datylonids and graduated cum laude with a dual degree in biology and German studies. After working briefly as a lab intern at the University of Wisconsin-Madison, Jill completed her PhD at UC Davis in the lab of Phil Ward, studying the evolution of ants. Her research interests include phylogenomics, global scale, historical biogeography, and taxonomy, particularly the resolution of cryptic species. In January, she will start a 3-year postdoc at the Senckenberg Museum of Natural History in Frankfurt, Germany, where she will turn her focus toward population genetics of Dorymyrmex ants and the evolution of social parasites.
Hello. I am not as animated or loud as Brendan, but that's hard to do. But I will still do my best to be enthusiastic and animated, even though I'm getting over cold myself. So, without further ado.
Share the screen first.
To kind of do a present review and come back.
Share it first. There's questions, right? Just look at the screen.
I think should we ask if people can see?
One second. I would love to have presenter. Alan, did you figure out the secret to that?
I don't think I have.
Okay. Well, give me 1 second to see if I can. That's the place.
It's Bob Anderson. Was that podcast again? It's Quentin Yoder's Center for Species Exploration. If you Google, I think, species exploration, Quentin Yoder, you'll get it. Yeah.
Oh, this will be a lot rockier if I don't have the notes. But I could potentially get through it without the notes. I would just greatly prefer to have them. So 30 more seconds to try that.
Oh, where it says sounds good.
Where?
There you go.
Okay, actually. Okay, give me one more second. I identified the problem.
But no stop point. You gotta keep the changes. If you could click, keep changing it. But yeah, you can't see it. That's fine. I mean, there we go.
No, wrong way around.
Oh, it's.
Sorry, we can see what's coming. Go back, there was a swap display. That's all.
I know, it's hard to see that main screen from there. Okay, got this. Thank you all so much for your patience. It'll be a more enjoyable talk if I figure this out, I promise. That one?
Yeah.
Yeah.
Oh, there we got it. Yeah, we can
You
see
got
just the one screen over here.
Okay, now it's a question of
Sharing.
sharing Zoom. All right, can I get confirmation from Zoom people that everything looks right?
Yes, does.
Okay. Then with that, I'm finally ready to roll. Thank you all. Okay. Hi, I'm Jill. I used to work IT and that was harrowing. And I recently completed my PhD at the University of California, working with
This one in Berkeley too.
excellent Dr. Phil Ward on the systematics of ants. So I'm here kind of last minute to give you an overview of what I did for my dissertation in the realm of species diversity, taxonomy, and modern methods in phylogeny in hopefully a really interesting and colorful way. So, title of my talk is UCE Phylogenomics Illuminates Remarkable Species Diversity and Clarifies Classical Taxonomy in Arid-Adapted Ants. This is the genus Dorymyrmex, and a wonderful little genus that can be recognized by this little point on the propodia. So, just briefly, I should convince you why ants are important, and actually of the utmost importance to life on the planet Earth. So we all care about insects. Insects make up about 80% of all known species. But among the insects, the biomass, 80% of it is ants. 20% is not ants. There are 20 quadrillion ants on the planet at last estimate. That is 2.5 million ants for every one of you. The ecosystem services they provide, the absolute power they have in making our world run is undeniable. So there's no surprise that there's a whole field of study about myrmecology. Can I hide that for people?
Yeah.
Still good? Great.
Yes.
Okay, so myrmecology is a broad enough range of research that it has its own named field of study. Ants are super diverse, iconic, recognizable. They have societies, they do agriculture, they're predators. If it's happening in animals, it's happening in the ants. Every single person on Earth has encountered ants, maybe, and there are over 14,000 known species, but we find more all the time. But some ants get more attention than others because, it's gonna load. Ants range in size considerably. So species that are tiny, difficult to access, and or less distinct can be relatively unknown. These are the extremes, of course. The ants you see on the ground are between these two, but this is the sort of diversity we're working with. This leads to an imbalance in taxonomic attention. The visually striking and easily diagnosable groups get most of it, but indistinct groups can contain just as much genetic diversity as groups that are flashier. These groups can often include cryptic species that have been independently evolving for millions of years, all without really changing their external appearance. One example of this phenomenon is the ant subfamily Dolichotorinae. These so-called odorous ants are chemically quite diverse and include the invasive Argentine ant, you may have heard of, that forms super colonies. They're also super smelly compared to blue cheese, banana Laffy Taffy, or rotting coconut, depending on the species and depending on your nose. That said, they're generally speaking medium small ants that have historically been really difficult to differentiate at the genus level, never mind the species level. So these indistinct groups need more work than their visibly diverse relatives do. All this is to say, there's so much we don't know about ants. Even things in our own backyards, there's undescribed biodiversity and undescribed species that await scientific ID, even in Phil Ward's backyard. Which brings me to the focus of my dissertation, this cute little genus, Dorymyrmex, which contains 61 valid species right now and is recognizable by the single medial cone on the propodium. Not a pair of spines, which is pretty common, but just one right in the middle like a party hat. We have no idea why. It's widespread in the Americas, and they're super common in dry habitats. Because they favor deserts, grasslands, scrublands, and beaches, they have this distribution that can be called amphitropical, or north and south of the tropics, concentrated in dry regions. And this is true, we think of the tropical regions, of course, as being the hotspots, right, for diversity and so on. And that's true for a lot of things, but not for Dorymyrmex. They're also conspicuous because they build these visible nests on open ground. Can you see me pointing? No. Okay.
Oh, there you go.
Yeah, yeah.
Oh, but now I can't see you pointing. Okay. So much for that. They build visible nests on open ground that look like craters or bullseyes. So they're a familiar sight to entomologists, and to my delight, they're frequently collected and easy to find. There are also at least 2 species of Dorymyrmex living in the Virginia, Maryland area. So these are really common ants, but we know almost nothing about them. One thing that makes them cool is they exhibit social parasitism. Some species are temporary social parasites of others, meaning one species will infiltrate the nest of another, kill the host queen, and chemically trick her offspring into rearing her own. Eventually, all the original colony dies, and the parasite colony is all that remains. They carry on their life cycle. The behavioral and chemical signals that they use to do this are still a mystery, and we aren't even sure how many parasitic species there are, although these are some of the possible relationships. But my postdoctoral research in part is going to explore some of these interesting social things. We also know that species with larger colony sizes, like Dorymyrmex bicolor, interact far more frequently with other ants, both aggressively with non-nestmates and affectionately with nestmates. My favorite thing about them is that they've been observed throwing little stones down neighboring ant nests in order to discourage their neighbors from foraging so they can go out and collect all the surrounding resources, which is wonderful. I think it's a little bit petty and oddly relatable, and it just gives them so much personality. But although they're pretty neat, they're still very poorly understood. And to ask broader, interesting questions about community interactions, pest management, social structure, behavior, we need to have a handle on the species definitions. So taxonomy has to come first. But taxonomy of Dorymyrmex has a chaotic past, and that's where I come in. So a brief history. In North America, taxonomy has been difficult because things largely look like this. There are many supposedly nondescript species. And in South America, well, it's also been difficult. They look a lot different, with a wide variety of forms that include spines and hairs and this crazy long semaphore, this basket under the chin, which is a special adaptation for carrying sand. The more homogeneous looking forms, like those in North America, are very widespread and can be found all the way down through Brazil and Uruguay. But these spiny striking ones, you can see the cone has elongated into a spine. These are restricted to the dry southern part of South America, but they're still united by having that single point process on the protein. So you might be thinking these seem pretty distinct, so the taxonomy must be easier. Maybe we can just focus on the fun South American taxonomy instead of the really difficult cryptic species in North America. But the world does not look like this with all South Americans. Proper systematic study of Dorymyrmex really has to cover the entire diversity of the group. So here we are. To resolve the tricky taxonomic problems of Dorymyrmex and discover its diversity both worldwide and in our own backyards, we have to start at the broadest level and work our way in. In turn, these little ants can teach us huge and consequential things about how the process of evolution unfolds, oftentimes unevenly, and they can even provide evidence for huge geological changes in Earth's history, like the timing of the Isthmus of Panama closure. Okay, chapter 1. Let's start at the beginning. This one's the least interesting, so I'll try to keep it short. The big questions are major lineages and relationships. What are they? How are they related to each other? Do modern methods corroborate traditional taxonomy? Does DNA confirm groups that were established using morphology long ago? Also, how old is this group? If they're really young, is that why they all look the same? Great, coming back to this slide, it seems like these species are very distinct, so the species level taxonomy should be easier, but historically the main issue for Dorymyrmex globally has actually been at the genus level. So are these southern species different enough to be relegated to their own genus, with a different one for the widespread species that are all similar, or should they all be considered part of Dorymyrmex? Well, the genus has been divided into as many as 7 genera and subgenera over the years based on these features. And in this diagram, each new color is a different genus. So the widespread similar looking species, including everything in North America, was this orange couple of subgenera here, Conomyrma and the subgenus Biconomyrma. In 1992, Steve Shattuck returned all of the species to the single genus Dorymyrmex, and which it doesn't seem too complicated at first glance, 7-to-1, whatever, but this is just one attempt of many, and this flowchart actually looks like this. So a taxonomic revision is going to be a lot of fun. Each vertical line is a published paper that changed the taxonomy of Dorymyrmex. And each color is a type of genus, and if there are multiple for a color, those are subgenera. So maybe Dorymyrmex isn't exactly understudied, because a lot of people have tried. It's just really complicated. So when Steve Shattuck lumped everything back together in 1992, he did so because of the cone, and based on that, the genus is unmistakable. This is where they've been ever since. Any divisions have been really uncertain. So I said, let's throw some DNA at the problem and see what happens. Very briefly, my methods are phylogenomics. So I'm determining these relationships with huge data sets of DNA sequences. I'm not going to dwell on this because you've recently heard several talks about UCEs and their magic. But basically, targeted genomics with ultra-conserved elements, aka UCEs, DNA extractions, targeting 2,500 loci instead of the usual handful for Sanger sequencing, data cleaning and sequence alignment, alignment being very important to ensure that homologous regions of the genome are being compared, and then partitioning and evolutionary modeling, which is where things are really the most important. So I've tried to model evolution as realistically as possible by doing a partitioning scheme I've abbreviated as SWSCEN, basically increasing model fit by chopping up the data into a bunch of partitions and then grouping similar parts of those together with the same model in order to make it more computationally tenable, putting like with like instead of keeping everything in the same order. I was doing my inference mostly in IQ tree exabays running on Cypress. Okay, so the results of these analyses show that 3 of the former genera hold up phylogenetically. So the green box here, the Dorymyrmex species group, used to be the Conamyrma. The Tenaire species group used to be called Dorymyrmex. And the orange group down there, the Flavescens species group, is Dorymyrmex sensu strictu, the ones with the spines. The horizontal length of the branches here indicates genetic distance. So these shorter branches are all extremely closely related. So this is a subclade here, the Dorymyrmex group, that shows extremely rapid evolution. To get a sense of just how rapid, we do a molecular clock analysis. I used MC tree. So basically, if we have the fossil age and the evolutionary rate based on the comparison to the other taxa in our analysis, we can triangulate the divergent states of the lineage splits. So I performed these analyses on a fixed topology. And here, these are the same samples, but the branch lengths represent time in millions of years. So the present is on the right. On each node is just the distribution of ages I estimated throughout the analysis. And here you can see there's also a general south to north pattern, the lines corresponding to the map. So that's kind of only by eyeballing, but we can see that the first lineages to emerge, let's see if I can point, are down here, which lead to southern South America, and then going further north as we scale up the phylogeny. The crown age of all Dorymyrmex is like 25 million years old, and the crown age of North American Dorymyrmex is part of that big schmear, that fast radiation around 5 million years ago. And these are basically mirroring what we see in morphological diversity. Bigger branches, more visually diverse. Shorter branches, really hard to tell apart. But the age of the Dorymyrmex group is much younger, so that all generally makes sense. Fix one thing here. Okay, so conclusions from chapter 1. What are the major evolutionary lineages of Dorymyrmex? There are 3 species. They originated in South America. That's pretty cool. Modern methods confirm that 3 of the former genera are actually phylogenetically sound. And Dorymyrmex is 25 million years old, but the ones that are undifferentiated are indeed quite young. So we have a good backbone, but the biogeography is mostly speculative. All of those were based on really sparse sampling. So I took it up a notch. Chapter 2, I did the same thing, but many more taxa. So I'm hoping to figure out, when did the widespread Dorymyrmex group arise in the South American overlap region? And how did Dorymyrmex migrate northward beyond the Amazon rainforest where it doesn't want to live? Chapter 2, phylogenomics, biogeography. Everybody knows about latitudinal diversity gradients. Generally, most people know the Amazon rainforest as a hotspot, which is a general trend is true, but there are some groups increasing in number that we know about that defy this paradigm, instead thriving in temperate or even polar climates. These are known as inverse rather than tropical heavy, or amphitropical or bimodal. But scientists are finding more and more of these so-called exceptions by the year. Other notable ones are Laria, creosote bush, another desert plant, and bees, broadly speaking, which paint a nice parallel to ants because they're, generally speaking, ground nesting and solitary, or have winged reproductives. Their notable group with an the cold is catching up with me. Another notable group with an amphitropical distribution is, of course, Dorymyrmex. So this map here is certainly undersampling Argentina and Chile as well, because the diversity there is really astounding. But it's pretty clear there's a kind of a bald spot over the Amazon basin. So faced with a distribution like this, we have to ask, how are these patterns produced and how and when organisms dispersed between North and South America? There are three main hypotheses for this large-scale American intercontinental dispersal. One being the Garlandia Caribbean land span, highly contested, but an option. The Isthmus of Panama, once land was connected, or long distance dispersal across the water, or some combination of all three. Luckily, Dorymyrmex are an ideal system to study these patterns because they're highly prevalent, frequently collected, capable of quick dispersal and colonization because they have these winged reproductives that can hop over the water. So, just like ground nesting bees. Big questions here. Are things recovered similarly when I expand by sampling? When did they develop this amphitropical distribution, and can we reconstruct their path? And what life history traits have enabled their dispersal? Methods, largely the same. A lot of my South American specimens were on loan from the Federal University of Paraná in Curitiba, Brazil. And without them, this wouldn't have been possible. So many thanks to Rodrigo Petosa and his lab there. Methods, largely the same. But here are the specimens I sampled, mapped onto approximate biomes. So, surprise, I found out there are actually 4 species groups, not 3. This Wolfhugeli group here in blue is new. The Paramicus group, Wolfhugeli group, generic group, and Flavescens group. I thought this Wolfhugeli group was just one aberrant species, but with increased sampling with others in the group, it shows how clearly the Paramicus and Wolfhugeli groups are divided. This is a relatively, this is a relatively long branch in terms of Dorymyrmex. But I never would have guessed it because the Paramicus and Wolfhugeli groups are basically indistinguishable micromorphology. Try as I might, there's no foolproof way to tell them apart, and they're both pretty visibly diverse, but they're definitely genetically divergent and sometimes geographically. You can see the Paramicus group is widespread, but the Wolfhugeli group is really relegated to the southern cone of South America and overlaps a lot with the Tenera and Flavescens groups. Here's a better illustration of that. Our little North American clade is nested amongst the Paramicus group there. And the map really mirrors the phylogeny in terms of south to north pathway. So more on biogeography in a minute. The molecular clock analysis shows that mostly we see the same thing. I'm going to walk you through this from the bottom. So main findings. Chronogram here, present is on the right again. The arrows here show that, firstly, the crown age of all Dorymyrmex is a little older, 27 million years old in the Oligocene, and the age of the crown Paramicus group, that big radiation, is about 12 million years in the middle of the Miocene. What matters is that long-term climate change probably helped drive Dorymyrmex diversification. The Oligocene, and especially the first half of the Miocene, were cool periods during which the grasslands and seasonal woodlands expanded. For example, this brighter yellow bar shows the early to mid-Miocene. And during this time, there was significant grassland expansion and the uplift of the Andes Mountains. So it reorganized the environmental conditions of the entirety of South America. Processes like these could have shaped the way Dorymyrmex thrives in dry regions, particularly those specialized southern taxa with weird adaptations, whose crown age is post-date this period. So scrolling up, we also see that the age of the Nearctic clade is somewhere between 10 and 6 million years. So Dorymyrmex reasonably reached North America between 10 and 6 million years ago. But just as before, the Paramicus group in general shows a rapid emergence of several major established lineages, kind of simultaneously, all over the Americas. So to dig into the historical migration, instead of just eyeballing it, like last time, I did some actual biogeographic analyses. I discretized the distribution into 8 regions and did a reconstruction in biogeobears, where I give it the distribution of each taxon and a phylogeny, and it tells me the possible geographic distributions of the ancestors at the nodes of the past. Here's another chronogram for results, but this one has colored nodes representing the inferred range of the ancestor at that node. So if it's a pie chart, these are percent likelihoods. Here we see from the arrows that the common ancestor of Borelius and Dorymyrmex, as well as the ancestors of the Flavescens and Tenere groups, all there in pink, stem from the southern cone regions. So this gray band, around 33 million years ago, represents the date of the hypothesized Garlandia Caribbean land span. So this is millions of years before Dorymyrmex had begun to diversify, never mind making it to the north end of the continent. So we can rule out Garlandia as a possibility for dispersal to North America. It's just too early. Let's look at the Dorymyrmex group. We see that actually Dorymyrmex arrived in North America just once and via the isthmus before it was finished. So the traditional date of the land bridge and the famous Great American Biotic Interchange is 3 million years ago, which is this right side red line here. But the green bar, oh, it's not green to you, is it? Yeah. Anyway, that green bar is the period of time when Dorymyrmex arrived in North America. So it appears that Dorymyrmex traversed the gap between the continents before the isthmus was actually closed. So they probably dispersed over water. Alternately, a growing number of scientists believe that the Isthmus of Panama story is earlier and more complex, with sections arising and shifting and submerging repeatedly, starting about 10 million years ago, which is the red line further to the left. So the asterisk over here marks the single point where the Dorymyrmex group has a dispersal from Peru to Baja California, which seems a bit extreme, Peru to Baja California. It seems to skip Central America entirely, but I think it's most likely an artifact of incomplete sampling rather than a jump dispersal. More on that in a second. So as for the Caribbean taxa, this was also interesting. They're nested within the South American clades. So there were multiple dispersals to the Caribbean islands from South America, but they never reached North America. Caribbean was sort of a dead end for them. The Central American taxa, though, are nested within the Nearctic clade. I sampled Central America hoping they would help illustrate that transition from South America to North America, but their closest relatives are actually in the Nearctic. There's at least one species found in Florida, Texas, and Yucatan, so there's some back and forth in the Gulf of Mexico region too. Basically, Dorymyrmex seems to be built for this quick range expansion, and my hypothesis is that it's because they're great at resource-poor environments, like deserts and scrublands and beaches, particularly beaches, because when you have islands that are shifting and submerging, if you're happy on a beach, you're going to be happy on any shifting island. So maybe they used shorelines as dispersal corridors. They've been found nesting at 1 meter above sea level in the Bahamas. So shifting islands seems like a pretty possible scenario. Inland, they have these really deep nests that help insulate them against extremes like temperatures and drought. So I think these same life history factors help explain the timing of Dorymyrmex dispersal to other groups. For example, I mentioned bees, ground nesting bees, but here we have, wow, the color change is bizarre. Okay, so this left bar here is the timing of Diadasia sunflower bees, estimated to have traveled from South America to North America during the Miocene, between 22 and 15 million years ago. Their biology, reproductively speaking, is kind of similar to Dorymyrmex, but this pulse of dispersal was too early for Dorymyrmex, I guess, to be far enough north. Dorymyrmex still is this bar here to the right. Another potential comparison is the turtle ants, genus Cephalotes, which colonized Central America several times, but we can't be exactly sure when. And I find this interesting because while Dorymyrmex and Diadasia bees are ground nesting, cephalotes are actually arboreal, so they may have had to wait for consistent shrubs and tree cover to appear before they could colonize new habitats. Anyway, it's fun to speculate about what the habitat might have been like on those Panamanian islands. It also barely predates the transit of Eciton army ants. So Eciton are like the super famous army ants that form the rivers of foragers in the forests. Anyway, they colonized Central America just after Dorymyrmex appears to have traveled. But this is interesting because Eciton doesn't have wingless queens. Eciton has these physigastric queens that are carried around by their workers. So they require solid ground in order to disperse. So Eciton and Dorymyrmex both provide further proof of Panama is actually older than its traditional age of 3 million years. So to wrap that up, there's a fourth major species group. There was one dispersal across the isthmus and some back and forth, and specializing on dry habitats has likely helped Dorymyrmex expand its range. Okay, now we haven't broached the question closest to home, the hairy question of Nearctic Dorymyrmex taxonomy. What's happening with all the Dorymyrmex that look the same in North America? So these are actually 5 different species. And taxonomy in North America has been so chaotic that Roy Snelling once said, "No North American ant has been cursed with such a singularly unfortunate taxonomic history as that described by Buckley as Dorymyrmex insanus." It's missing the type material, there was a neotype designated, there was a new neotype designated, and that's only one species. For nearly a century, the only system we had was there's a bicolored one, a dark one, and a yellow one. Plus one that doesn't count because it's only known from the type series. There were some major additions by James Traeger, Roy Snelling, and Mark Dayrup, which led us to 13 species as of 2017 when I started my PhD. But some researchers have long suspected there are more, or just some crazy variation among them. Unfortunately, I don't know how different these look to you, but these look very different to me. What I want you to focus on are the head width and the concave vertex of the head. Basically, this is within colony variation. These are 2 paratypes of Dorymyrmex smithii. And the same characters that are useful for differentiating species are the same ones that vary within a single nest, never mind within a single species. So I think some of it is just allometry, and you have to look at the largest workers from any colony in order to find these differences. But the question is whether there are fewer species in North America that show a lot of variance or just more species than we know about. But with the exception of a few complexes, there's hope for using morphology. Because of the heads, by integrating molecules, morphology, and geography, we can actually start to pick these things apart. So if we use an evolutionary tree generated with molecular data as a framework, we can then find diagnostic features to, provide some sort of working key to this group, so you don't have to sequence everything. Unless you're sure. The big questions here are how many species of Dorymyrmex really exist in North America? How are they related? And can we distinguish them using worker morphology? Methods, we're maximizing taxon sampling as much as possible, but I really need more from the American Midwest. They aren't so much in the Appalachians, but all of the Plains states, they gotta be there. But to start the process, there's expanded taxon sampling, and then I did a bunch of morphometrics, such as head width and length, interocular distance, the percentage of the head that is behind the eyes, like all of these really obscure kind of proportions, as well as some profile views of the mesosoma. So there are different profiles of the mesonotum and profiles of the propodium and the vertex of the head. These all seem really mushy like they grade into each other, and they do, but it's the best that we've got. So I did 24 measurements, calculated 22 ratios, measured 148 specimens, and made a phylogeny. So let's start with that. Very short branches at the base of this tree, but there are 4 major clades that are maximally supported. The first of these hints at southern ancestral range, 2 new species. The second of these is large with relatively wide heads, and these are found in Nevada and Utah, but also California, but also Florida, but also everywhere, but also Arizona. Clade C here. There are 8 species, 6 of which are new. These are really diverse in size, shape, and color. So we assume they're all good species. Some of them have been observed doing different things ecologically as well, which I don't have time to go into, but these are more Western groups, California, Oregon, Baja California, Nevada. And then clade D, with 14 species, have smaller, narrower heads, and this is another clade that really spans North America. There's a big Floridian radiation, as well as things in California and Arizona, and even Honduras popped in there. That was one of those Central American taxa nested within the Nearctic clade. So, to accompany this, I developed a set of 54 figure plates as part of my dissertation to illustrate these morphospecies in lateral view, dorsal view, and the individual variation, and drew up a provisional identification key to them. Identification of Dorymyrmex workers is difficult, but not impossible, although some of the couplets do eventually just lead to blah, blah complex. But this is only thanks to the framework established using DNA. What really became striking when completing this work is that the convergent evolution of color in Dorymyrmex is rampant. So what people have been calling Dorymyrmex bicolored one in the Western US, is actually any one of 5 lineages. And Insanus could be any one of 4 lineages, but Insanus is also the wastebasket taxon for the dark brown. In the future, so during the rest of my time here and also at the Senckenberg Museum, I need to examine the queens and males, increase my sampling of Arizona and Mexico, where there's crazy diversity, and put together that formal taxonomic revision. The queens and males are important because they're super closely tied to evolution, being the reproductive parts of the colony, right? But they're so rarely collected along with the workers because they're more difficult to access, or your timing has to be great. My third chapter was going to be a systematic revision, but I didn't quite expect to double the number of species in North America, and I want to do a good job with it. So, to sum that up, how many species in North America? 27 and counting. I just got a bunch of new sequence data back from the Ward Lab, and I'm excited to dig into that. Similar phenotypes have evolved multiple times in parallel, creating a lot of the confusion that plagued taxonomy. But now that I know there are 5 bicolored lineages, I can start to pick them apart. And they're usually distinguishable using worker morphology, but don't test my key yet. I'm still working. Ultimately, this whole PhD presentation wasn't just about ants. What I really care about is getting to know the living world around us, resolving old problems and technologies, learning how evolution creates change asymmetrically, and finding further events and evidence for them in Earth's history. Before I finish, I need to thank many people, institutions, funding agencies, Bill Ward, Rodrigo Petosa, people who have been wonderful hosts to me, colleagues, collectors, conversationalists galore. I want to shout out my other ongoing projects, this revision and evolution of social parasitism with Christian Ridling, Bob Johnson, and Steph Kober, and evolutionary history of the subfamily Dolichotoridae with my former lab mate Zib Lieberman. Here's my information. I'll put that back up in a second, and I'm happy to take any questions you have. Thank you.
That was great. Thanks a lot. What do they do when they eat? Are they sort of omnivores?
Yeah, they're generalist scavengers, basically. They really like to tend nectarines and will also tend lycaenid larvae, and visit extrafloral nectaries, that sort of thing. You could probably find them on peony buds. Whatever they can find.
Another ecological question, when you showed the burrow and you said it was deep, how?
Depends on where they are. They're not that deep in the Bahamas, but in Florida, inland Florida, they can be 2+ meters deep, just a single channel going straight down. Walter Schinkel has a great book on ant architecture, where he explains some of the experiments he's done testing the humidity and the different air composition at various depths in an ant nest. And it's absolutely fascinating.
How does he do that?
I can't recount those methods here, but lots of creating artificial nests and adding sensors into where he knows the chambers are. That's
Okay, then about the dispersal through maybe the putative islands of the isthmus area. So the army ants have non-flighted queens, right?
Yeah.
And then your group has winged ones. What about just driftwood? I mean, we're just talking about one event here. I mean, a few million years, islands dancing around, couldn't that have happened, or?
Sure. Yes.
Okay.
I think it's likely that Dorymyrmex moved by flight, but I think it's likely that Essaton had actual land, because you're not going to get an entire colony of army ants with millions of workers and their queen on a little piece of driftwood moving into South America. And I'm pretty sure that dispersal happened a couple of times. But don't quote me on that last part.
Nobody can check you anyway.
In your chapter 2, you have a distribution map with some dots on some islands away from the coast. How did these ants get there? Is it like a more recent introduction, or yeah? So
that's a great question. So let me see if I can go back. Not very elegant. So those were likely points that I did not sample. These are the ones that I did sample, so really no.
Okay, but on another previous one, you have a black dot, yeah.
Right, so to clarify, these are my chapter 2 specimen sample, but here, this distribution of Dorymyrmex based on records on AntWeb and other databases. There are Dorymyrmex in the Galapagos. This is probably a relatively recent dispersal because they look just like the ones in Ecuador, the mainland Ecuador. I haven't sequenced them. I would like to. Same goes for off the coast of Mexico here. I haven't sequenced them, but they find their way around.
That one off Mexico, there's been a lot of guano mining, could easily have come in on a ship.
Oh, definitely. I think that is probably true of the Galapagos as well. There's been a lot of that. But I don't know just how young they are. The Caribbean island divergences are a million years ago, so probably not.
It's super fascinating that you're estimating the ages of geological features based on phylogenies at which they're emergent. That is like really fascinating confluence, different kinds of sites. If you can argue with geologists, they're like, no, it had to have been this many million years ago. Like, no, then my data said this many million years ago. Like, who like how are you going to say that your data is better?
Yeah, so the study that I sort of watched over briefly about somewhere else, but about the the more complex and earlier dispersal theory by Bacon et al. 2017, they took into account terrestrial fauna, they took into account marine fauna. They did involve some geology, although I don't recall that bit because I wasn't so interested in the rocks. But they did used multiple lines of evidence to demonstrate pretty convincingly that this is older and probably happened in a more complex way than we've been thinking about.
I'm curious about the whole Wakely group.
Wolfugli.
Wolfugli. You mentioned that it was kind of an unexpected clade.
Yeah.
That you thought it was just one single variant. I'm curious, scientifically, how did you pursue that? Why did it seem worth sampling more? What was the hint that there was something more to that group?
Right. So the hint that there was something more to that group was really just the length of the branches in the phylogeny. So here we have that phylogeny, where's my pointer?
Okay, so it's those two.
So it's just these two right here. I was like, those look exactly like the others. Why are they so different? Maybe if I sample further, I'll find more on this branch right here to help illuminate the question of this emergence. And so I sampled more of the things that look like all of this so-called Dorymyrmex group. It turns out that some of them were in a different species group entirely, and I just couldn't know that. It wasn't on purpose, basically.
Right. I like to hear about the process. So did they come from a weird place, or was there anything other telltale about them?
Really, no. And that's why I was wondering why the heck they were so distant from the others.
All right.
But they were literally on the street in Curitiba, Brazil. I collected them on my way between my Airbnb and the university. They were on next to a road. I picked them up, and the Wolfugli group came into being.
They're not part of that blue area in the map, are they? Like the dry charcoal?
Yeah.
Oh, yeah. So dry charcoal, like a very dry charcoal, or like the dry diagonal may have an influence on those guys.
Yeah.
Going on with the big hole in the middle of Brazil, you talked a lot about how things are going up from South America into North America, Central America. Is there something that's keeping them out of that chunk of not?
Yeah, so this has been the most, that blank white spot there in the Amazon basin has been the most consistently rainforest chunk of South America over millions and millions of years. And for whatever reason, Dorymyrmex has just never latched onto that. So I think during the contractions and expansions of the grasslands, as the rainforests shrank, even if they didn't shrink, Dorymyrmex has been able to use coastlines to get around seemingly big barriers like the Amazon basin and the rainforests of Central America, because they can just kind of camp out on shorelines. My hypothesis.
Well, along those lines, it didn't seem like you had many samples from the
trees.
So just lack of sampling.
Yeah, basically lack of sampling.
So the Inter-Andean Valley has been dry. I think they would.
Absolutely very dry. We have this one right here. So here is the what's up? So up here at the top, the asterisk is where we see that jump from Peru to Baja California. And in Dorymyrmex radiation terms, this is a relatively long branch. So I imagine there's more happening in that western part of South America that I just don't have the sampling for. This one Peruvian specimen, Phil Ward collected in like 1979. But if you know any Peruvian myrmecologists, please give me their.
So given the fact that this group favors arid environments, one might not expect them to show up in amber. Do they have any kind of fossil record at all?
That's a great question. I'm still waiting for them to find a Dorymyrmex in amber. They found a Forelius, which is the sister genus that you keep seeing in my outgroups here. Good. Yeah, this down here. There's a Forelius fossil, 2 of them now, but no Dorymyrmex. So I think the issue with this is that the Dominican amber fauna, which has been sampled or generally observed relatively thoroughly, and is my impression. That's about 15 million years old, and Dorymyrmex wasn't up there yet. Based on my divergence stating, they were still, down in South America at that point. So they don't show up in Dominican amber. The Forelius show up in Chiapas amber from Mexico, and that's older though too. That's like 23 million years. So I think Forelius somehow moved north earlier than Dorymyrmex and ends up in Chiapas amber, or it's some undifferentiated dolichotorine ant that looks a lot like a Forelius that isn't. Or yeah, I don't know, but that's also too early for Dorymyrmex. So I don't know of any good South American deposits, but I haven't found any great Dorymyrmex fossils or any, Dorymyrmex fossils, but I'd love some so that I can calibrate my chronograms.
Anybody else has
Oh,
questions on one?
good question.
Raise their hands.
There.
If you go up to the top of the screen, as the bar drops down.
Oh, sure. So, it does, I'm gonna open back up. Are you also in Zoom? Can you look at the chat?
Yeah.
Okay. Hello, chat people, virtual people. Thanks for coming.
But I understand right, like the most of the groups down in the southern South America haven't really great people that Sahara area.
Yeah.
Surprised
Yeah.
stuff. I assume that's been pretty well seen.
Yeah, sort of. So
Mitosis has done a whole lot.
yeah. In general, the dry diagonal of South America, the Sahara, is not as well controlled as the rainforest, because people just haven't been as interested in it. This rainforest is a diverse place. But the southern, the crazy looking taxa, the Teneric pubescens groups are, I think, just such extreme specialists that they, since their common ancestor with the other Dorymyrmex, they've just specialized so much on the southern environments, the really dry Patagonia and west of the Andes, and then northern part of the, going up into Peru, the Atacama. They just, I don't know, they're optimized for sand life, basically. Although it's drier in Brazil, it's not that.
So are you recognizing back to reverting to the genus, with the ones with the specialized sand carrying? So you were kind of related them back to the genus that they were recognized as before, or?
I haven't made any drastic genus level changes.
Oh, okay.
I've gone as far as saying they're phylogenetically sound.
Okay, yeah.
Monophyletic and robustly so, but I still don't feel qualified to
really. Immediately,
I should probably take charge.
When you said that, I was like, "Whoa, that's like a totally new genus." Yeah. I mean, to have that specialization of carrying the sand, like a pouch or whatever. That's crazy.
Then they'd be the real Dorymyrmex. What do I call these other ones?
Ah, that's awesome. That's part of what we do, right?
Yeah.
From a diagnostic standpoint, how does the barcode perform within Dorymyrmex?
Good question. It's been a couple of years since I did any barcode, like single gene analysis, but what I did notice just pulling out CO1 as bycatch from my UCE data, is that it has helped pull apart some of the really short branches within the Dorymyrmex group, but the topology is also less certain. So I think there is, you know, different genes that are evolving differently, and having the full UCE complement is helpful to figure out the consensus.
It's always that kind of balance from diagnostic standpoint. I mean, sometimes I'm too interested in the phylogeny of the barcode, but if they can tell me, is it this or that? But even that sometimes is like, well, that's not, I'm not happy with the support.
Yeah.
What's the person who's like patient enough to watch?
I know. I was gonna ask that. I don't
Come on,
remember.
once you start watching,
It's
it's
probably
really
one of
hard to
those-
stop.
Ant farms. That's what I thought. I was like, find somebody with an ant farm.
This was out in the field. They're different genera entirely.
I thought maybe the Budno had been watching them, you know.
Seems likely. This is back in 1979.
What's that called from German? Freud?
Schadenfreude.
Schadenfreude.
Watching, taking pleasure in others' misfortune. Something like that. You would think so, but there's less direct confrontation this way. I think that's why it appeals to me. There's no face-on interaction. You're just making it more difficult for them.
Fine. Great. Cool. Well.
Thank you.
Thank you so much.
Thanks again, Joe, for stepping up at the
Yeah,
last minute.
absolutely.
Fantastic.
It was no way fortuitously. That's
That's
Yeah,
right.
That's
That feels-
Oh, yeah.
You know, he is online.
Too bad it's still.
He's usually louder than me, so without him here, I can interact with you.
Shut down.
Are there any more comments, questions, announcements before we close 1,239th meeting in South Washington? Having heard nothing, I have a motion to close said meeting.
So moved.
Seconded? Second. Thank you, everyone. See you next time.